Hello, everyone. We will now begin OliX's first online IR briefing for 2026. Before we get started, I'd like to sincerely thank all of our shareholders and viewers for your continued support. Today's session is designed to give you a more detailed update on our R&D progress and business strategy. Let us introduce today's presenters.
This is D.K. Lee, Chief Executive Officer at OliX.
This is Dr. Jun-Hyun Park, and I am in charge of Head of Research.
I am Young-Jin Kim. I am in charge of Finance Part.
This is Kelly Kim, and I am in charge of Business Development.
I am Han Sok Ko, and I am responsible for Corporate Strategy.
D.K. Lee, Chief Executive Officer at OliX, will present the newly defined growth strategy under the OliX 2.0 roadmap and introduce the new name of our platform technology.
Hello, I'm D.K. Lee, CEO of OliX Pharmaceuticals. Thank you for joining us today. I would like to once again express my sincere appreciation to all of you for taking the time to join and watch this briefing. Let me begin with a brief introduction to our company. OliX was founded in 2010 with the mission to improve human health and quality of life through the research and development of RNA interference therapeutics, and listed on Korea Securities Dealers Automated Quotations in 2018. As of now, approximately 96 employees across our headquarters in Korea and our subsidiaries in the United States and Australia are working together. In particular, the past few years have been a period of meaningful inflection for OliX. To date, we have entered into 5 global partnerships, demonstrating the competitiveness of our technology, and in 2025 collaborations with Eli Lilly, a leading global pharmaceutical company, and L'Oréal, a global leader in the beauty industry, further validated the value and scalability of our technology in the global market. We view these achievements not merely as business milestones, but as a critical starting point for the global expansion of our RNA-based therapeutic platform. Today, we will provide a direct update to our shareholders and market participants on OliX's recent business operations and R&D progress, and, based on this, share our future growth strategy and strategic direction in a more transparent and concrete manner. To better communicate this direction, we've redefined our growth strategy and introduced a new name for our RNAi platform, which I'll walk you through now. Let me walk you through OliX's new growth strategy. We recently established the OliX 2.0 roadmap, where we defined our next-generation R&D platform strategy and expanded both our corporate identity and overall growth strategy. The name OliX comes from a combination of oligo, referring to oligonucleotide-based therapeutics, and the idea of accelerate. It reflects our original vision to accelerate drug development by combining world-class oligonucleotide technology with a pragmatic approach to target selection. In that sense, OliX 1.0 was really about accelerating drug development based on RNA interference technology. And now, we're moving into the next phase. With OliX 2.0, we're evolving our core identity from accelerate to excellence. So rather than just focusing on speed, we're now aiming for excellence across the board, from R&D and platform technology to pipeline strategy and global partnerships. And to better reflect this direction, I'd like to introduce, for the first time, the new name of our RNAi platform. The siRNA industry has, until recently, focused on validating technological risks within a relatively limited set of applications, thereby laying its foundational groundwork. But now, With those technologies validated, we're seeing rapid expansion into a much broader range of diseases and delivery approaches. In this environment, we believe it's important to move beyond a single technology and build a scalable siRNA development system. And that means bringing together different technologies and delivery strategies into one unified platform. Based on this direction, we'd like to introduce the new name of our siRNA platform, OASIS. OASIS stands for OliX Advanced Small Interfering RNA System. The name OASIS embodies OliX's commitment to offering new therapeutic possibilities and hope to patients, much like an oasis providing life-sustaining water in the desert, symbolizing the therapeutic value and corporate identity that OliX aspires to uphold. Through OASIS, we intend to rapidly broaden the application landscape of siRNA technologies and accelerate the development of next-generation RNAi therapeutics.
Here, you can see the current clinical development status of our key pipelines built on the OASIS platform, as well as our future development plans for these programs.
Let me begin with OliX 702A, our pipeline candidate for the treatment of MASH and obesity. OliX 702A is currently in a Phase 1 clinical trial in Australia. The single ascending dose study began with first patient dosing in February 2024, and the last SAD patient visit was completed in February 2025. We are now in the final stage of the multiple ascending dose study, with the last MAD patient visit expected for April 2026. Although disclosure of the most recent clinical data is limited due to our agreement with partner, we have observed an average liver fat reduction of approximately 60–80% in the SAD cohorts, and this effect has been shown to persist for more than 10 months. We consider these data to be clinically significant, as they indicate a sustained and potentially best-in-class reduction in liver fat. Once the Phase 1 study is completed, we expect our partner Eli Lilly to take the lead the subsequent Phase 2 development. Through this collaboration, we anticipate further acceleration in the development of OliX 702A and enhanced global development potential. I will now discuss the clinical development status of OliX 104C, our pipeline candidate for the treatment of hair loss. OliX 104C is an siRNA-based therapeutic that is administered locally to the affected areas of hair loss. When administered locally, the drug achieves high concentrations in the scalp, and, due to the intrinsic properties of siRNA, any portion that enters the systemic circulation is cleared relatively quickly. These characteristics offer the advantage of ensuring systemic safety while maintaining pharmacological activity at the local site of administration. In January last year, OliX 104C demonstrated a favorable safety profile in a Phase 1A clinical trial conducted in Australia. And, following approval of the Phase 1B and 2A clinical trial protocol, a Phase 1B study is currently underway in Australia. The Phase 1B study is designed to evaluate safety and tolerability under repeated dosing and is targeted for completion in 2026, followed by a Phase 2A study aimed at evaluating efficacy, which we plan to complete in 2027. By pursuing a strategy of accelerated clinical development, we expect to strengthen our leverage in future licensing discussions. Lastly, let me discuss OliX 301A, our pipeline candidate for the treatment of age-related macular degeneration. In the preceding Phase 1 clinical trial, we assessed an excellent safety profile in both single-dose and multiple-dose studies and observed potential efficacy signals such as improvements in visual acuity in several patients, including those with geographic atrophy. We are currently engaged in various discussions with multiple global companies regarding OliX 301A, and in order to further enhance the asset's value and strengthen our future licensing negotiation power, we are preparing a Phase 2A clinical trial in patients with dry AMD.
Next, Kelly Kim, Director of Business Development, will walk you through the status of our ongoing partnerships.
Hello, and thank you for joining us again. I'm Kelly Kim from the BD team. I'd like to first mention that we've been hearing a lot lately that the RNA era has begun. I believe this really reflects the growing interest in our company as well. We've been working hard to meet those expectations, and there are some meaningful BD updates that I'd like to share today. Why don't I start off with giving you a quick update on the partnerships that are currently ongoing. First up is OliX 702A and the partnership we have with Lilly, which I believe has been getting a lot of attention in the market. As briefly mentioned by DK, OliX 702A is currently in Phase 1B clinical trial in Australia. Since partnering with Lilly last year, we've been actively discussing the study, and we're now working closely to minimize development risks and accelerate the program to the next phase. So far, the preclinical and clinical data support developing OliX 702A as a long-acting formulation with the goal of at least biannual dosing. And we expect this long-acting profile to become a key differentiator within the MASH and metabolic disease markets. As many of you are aware, Madrigal's MASH drug, Rezdiffra, has been generating solid annual sales of around $1 billion since its launch. We think this really highlights the nature of the MASH market. Since MASH is a chronic condition, it's an area where once efficacy and safety are confirmed by doctors and patients, therapy typically moves into long-term use. Given that Rezdiffra requires daily administration, and Wegovy is administered once weekly, we believe OliX 702A's long-acting profile offers a clear point of differentiation. And in fact, the duration of efficacy is something Lilly sees as a key attribute. Furthermore, major pharma like Pfizer are actively pursuing long-acting obesity treatments, highlighting the growing interest in this space. In line with this trend, OliX also has seen in animal models that combining OliX 702A with a GLP-1 drug gives extra weight loss and some additional synergistic effects. Based on this, it's reasonable to speculate that Lilly could potentially expand 702A's indications to include MASH and other metabolic conditions. In addition, the agreement with Lilly isn't limited to the Marcone target alone. It also includes an option for Lilly to exercise if we move forward with dual siRNA development, incorporating additional targets, and discussions around this are currently ongoing. We'll have Dr. Junhyun Park, our research head, provide more details on this dual targeting platform shortly. Great collaborative efforts are happening behind the scenes in this partnership, though we can't share much details just yet. With L'Oréal, a collaboration under the theme of skin and hair regeneration and longevity is ongoing. As a global leader in the beauty industry, L'Oréal is providing practical R&D support across the collaborative programs. As a result, the programs are progressing smoothly according to the planned timelines. Our partnership with L'Oréal is going very well. Since our collaboration spans both skin and hair programs, we will be sure to share any exciting news as soon as it becomes available. In addition, our long-standing partnership with Hansoh Pharma continues to move in a positive direction. I personally see Hansoh as a key partner that has helped lay the foundation for our global licensing and collaboration efforts. Hansoh is a leading global pharmaceutical company in the oncology and cardiovascular metabolic space. As one of China's major pharma players, they are active not only in advancing its own pipeline, but also in both in-licensing and out-licensing. With the rapid growth of China's biosector in recent years, Hansoh's position in the global market, which was already strong, continues to strengthen even further. Hansoh has already exercised additional options, and we continue to work closely together under a strong partnership, advancing both the programs and the underlying platform. As development progresses, we see potential for additional milestones as well as a range of further collaboration opportunities. As you can see, there are multiple opportunities, even within our existing partnerships. As these active discussions continue, we look forward to sharing updates if there's any expansion of the collaboration or other meaningful progress.
We will now invite Dr. Jun-Hyun Park, Head of Research, to present the R&D status of the OliX 2.0 programs. First, we will provide an overview of the R&D status of the OliX 501A program.
I would now like to discuss OliX 501A, our program targeting ALK7. Let me first explain why we are focusing on adipose tissue as our target tissue, primarily because we aim to address obesity. As you may know, among current siRNA delivery technologies, the GalAC platform is highly effective in targeting the liver. However, targeting the liver alone has limitations when it comes to addressing obesity at its root. Obesity is not merely a state of excess fat accumulation, but in many respects can be understood as a dysfunction of adipose tissue. Adipose tissue is not just a storage depot for energy. It also serves as an important endocrine organ that helps regulate the body's metabolic balance. When adipose tissue function is impaired, this can lead not only to obesity, but also to a range of conditions such as diabetes, fatty liver disease, and cardiovascular disorders. As you are well aware, the current obesity treatment market is dominated by GLP-1 agents that reduce body weight primarily through appetite suppression, whereas our approach focuses on more fundamentally improving adipose tissue metabolism. To this end, our development target is a gene known as ALK7. ALK7 functions as a brake on lipolysis in adipocytes, and by using our siRNA technology to inhibit ALK7, this is expected to promote fat breakdown. Accordingly, this strategy is aimed not only at reducing body weight, but also at improving adipose tissue function in itself. And ALK7 can be viewed as a target with the potential to deliver synergy when combined with GLP-1 therapies. This slide describes our adipose tissue targeting platform, OASIS or Adipose. We have identified a proprietary lipid ligand that enables the selective delivery of siRNA to adipose tissue. Through extensive screening, we discovered siRNA structures and chemical modifications optimized for adipose delivery, and we have completed patent filings for these innovations. As shown in the data below, in mouse models we confirmed that subcutaneously administered siRNA is delivered efficiently to adipose tissue, while having minimal impact on other organs such as the liver and kidneys. On the right, you can see that a single 3 mg per kilogram dose in mice achieved more than 80% target knockdown, and this effect was maintained for over 2 months. As mentioned earlier, we have designated OliX 501A as the codename for this ALK7 targeting program. These data represent proof of concept results obtained in obese mouse models, and the compound will undergo further optimization to enhance its competitive profile. In obese mouse models, an administration of this early-stage compound led to a statistically significant body weight reduction of approximately 10% at day 28. More importantly, looking at the qualitative aspects from the body composition analysis, at week 4, fat mass was reduced by more than 30% compared with controls, whereas lean mass did not decrease and in fact showed an increase. In other words, the weight loss was driven primarily by a reduction in fat mass without loss of muscle, highlighting why inhibition of the ALK7 target represents a compelling therapeutic strategy. These data show the results of combination dosing with Zepbound. In general, when Zepbound is used in combination therapy, its dose is often reduced as a strategy to mitigate adverse events. It is therefore important to determine whether weight loss efficacy can be maintained at the reduced dose, and whether side effects such as loss of lean mass can be alleviated. With these points in mind, we evaluated the potential of combining ALK7 siRNA with Zepbound. In obese mouse models, we compared a high-dose Zepbound monotherapy group with a combination group in which the Zepbound dose was reduced to 1/10, with OliX 501A added on top. As shown in the data on the left, in the low-dose combination group, the degree of fat mass reduction was maintained at a similar level, and, after treatment cessation, weight rebound was markedly attenuated. On the right, you can see that loss of lean mass was markedly reduced in the low-dose combination group. This slide presents the efficacy data for OliX 501A in non-human primate models. First of all, the compound used in these data is not a fully optimized candidate, but an early lead, and the purpose of this study was to confirm whether this sequence is actually active in primates. Following a single administration at a dose of 3 mg per kilogram, we observed up to an 84% reduction in ALK7 mRNA in adipose tissue at the 2-week time point, with approximately 70% suppression still maintained at 4 weeks. Importantly, despite using an early, non-optimized compound in this study, we were able to demonstrate efficacy that is comparable to competing agents. We are now advancing an optimized version of OliX 501A with further improved potency and duration of effect. The data you see here are from our recently established optimized platform. We have continuously refined the lipid modification scaffold and conjugation sites from the initial platform, and as shown in the graph on the left, under a single low-dose administration, the optimized compound demonstrates superior activity compared with the original lead. When this optimized platform was evaluated in obese mouse models, the magnitude of body weight reduction increased substantially, which also translated into marked improvements in body composition parameters. We believe this profile is at least on par with, and potentially competitive as a best-in-class candidate relative to competitors' ALK7 program. Following completion of preclinical proof of concept studies in animal models, we are targeting an IND submission in the first half of 2027. As ALK7 is already a clinically validated target, we believe that, provided we confirm sufficient efficacy and durability in non-human primates, there is meaningful potential for early out-licensing opportunities in preclinical stage. With our adipose tissue delivery platform now in place, we are actively screening a range of additional obesity-related target genes beyond ALK7. We expect to secure new obesity pipelines beyond ALK7 in the near future.
Next, we will provide an overview of the R&D status of the dual target platform.
To share some of our recent work on dual targeting, we have now established a dual target siRNA platform for ocular indications in addition to our GalAC-based platform. Dual target strategies are particularly important in ophthalmology because many ocular diseases are not driven by a single cause, but are complex disorders in which multiple mechanisms, such as inflammation, neovascularization, neurodegeneration, and metabolic stress, act in parallel. Our goal is therefore to achieve a more comprehensive therapeutic effect by modulating these pathways simultaneously. Intravitreal injections are highly invasive and place a significant burden on patients, so being able to inhibit two targets with a single injection represents a major advantage in terms of patient convenience. To this end, we have made extensive efforts to develop the appropriate construct, which, as illustrated, consists of two siRNAs linked together via a linker, with a newly identified lipid ligand, discovered through screening, conjugated to this dual siRNA structure. First of all, it is technically more challenging to deliver siRNA to ocular tissues than to many other organs. We therefore screened a wide range of chemical modifications, linker structures, and lipid ligand combinations, and through this work we identified a lead substance and generated meaningful data. As shown in the data below, after a single intravitreal injection and analysis at day 7, the dual target molecule produced a significant reduction in the expression of both targets. We also confirmed that this dual target construct achieved a stronger inhibitory effect than the combination of two separate single target siRNAs. To our knowledge, this is the first time in our field that dual target suppression has been demonstrated in ocular tissue, which we consider highly significant. We believe we have now established a dual target platform for ocular delivery, and going forward, we plan to select pairs of targets that are well suited for indications such as GA and other ophthalmic diseases and advance them into development. This section revisits our GalAC-based dual siRNA platform, which we introduced previously. As with our ocular dual platform, the key advantage of the GalAC dual platform is that instead of administering two separate agents, we can design a single molecular entity that simultaneously and uniformly suppresses two genes within hepatocytes. This approach reduces variability compared with simple combination dosing, and it is also more straightforward from a CMC standpoint, which in turn improves overall development efficiency. We have now completed optimization of the structural design and chemical modifications, and we believe the platform offers sufficiently competitive potency. These are the most recent data we have obtained on this platform. After establishing the dual target platform, we conducted studies on multiple target pairs to determine which combinations can be co-targeted effectively and exhibit synergy. While we are not disclosing the specific targets at this time, these data are from a combination of two metabolic disease targets, A and B, comparing single-agent dosing with combination dosing. The results show that, under combination dosing, both targets are simultaneously suppressed without mutual interference, and we observed knockdown at least comparable to monotherapy not only at the hepatic mRNA level but also at the protein level in blood. We then evaluated how this target combination affects metabolic parameters, and among the various endpoints, we observed particularly strong effects on triglyceride-related markers. First, as shown in the data on the left, plasma triglyceride levels were significantly reduced in the combination group compared to single-agent treatment. A similar pattern was observed in the liver, where hepatic triglycerides were reduced to the greatest extent in the combination group. These findings suggest that the combination has the potential to lower triglycerides more effectively than existing therapies. Based on these results, we have initiated the development of a dual target candidate based on this combination.
Finally, we will proceed with an update on the research status of our CNS-targeted platform.
As many of you know, the biggest hurdle in developing therapies for neurological diseases is the blood-brain barrier, or BBB. The BBB fundamentally serves to protect the brain from external substances, but from a therapeutic standpoint, it becomes the major barrier. And even though siRNA molecules are smaller than antibodies, they likewise face significant difficulty in penetrating the BBB. In the siRNA field, intrathecal or other direct CSF injections are sometimes used to bypass this barrier, but these routes are inherently invasive and place a substantial burden on patients. In light of these needs, the prevailing industry view is that having delivery vehicles capable of penetrating the BBB has become essential for next-generation CNS therapeutics. For this reason, we have sought to secure a transferrin receptor, or TfR, shuttle, which is currently one of the most widely recognized BBB shuttles. To briefly explain, transferrin is a protein that transports essential iron throughout the body, and because the brain also requires a constant iron supply to maintain normal function, brain microvascular endothelial cells express high levels of TfR. By exploiting this pathway, we can conjugate our siRNA to a TfR-binding protein, enabling receptor-mediated delivery into the BBB. As illustrated in the figure on the lower left, global pharmaceutical companies including Roche have already generated meaningful brain delivery data using this TfR-based strategy. This slide provides a high-level overview of the competitive landscape for TfR-based development programs. First, in the antibody-based space, major players such as Roche and Denali have advanced TfR shuttle antibodies for Alzheimer's disease into late-stage clinical development, whereas nucleic acid therapeutics are still at an earlier stage. Most nucleic acid programs still rely on intrathecal administration, and on the siRNA side, Arrowhead is emerging as a frontrunner applying a TfR-based platform. But overall, the field remains in an early phase. In this context, we believe that securing a TfR-based platform presents significant opportunities for OliX as well. When we evaluated various TfR1 shuttles, we concluded that the format best suited to our siRNA platform is the antibody derived from camelid species. Conventional antibodies are relatively large, at around 150 kilodaltons, whereas camelid antibodies have a simpler structure and a smaller size. Among these, the isolated antigen binding region, shown in blue here, is known as a single-domain antibody, or VHH, with a size of roughly 15 kilodaltons, which is highly advantageous for BBB penetration due to its small molecular weight. Its inherently simple architecture also makes it amenable to further engineering and optimization. The non-human primate data on the right are from Ionis, which evaluated Vectorus's TfR1 VHH shuttle, now the basis of our collaboration, conjugated to siRNA and showed meaningful target knockdown not only in the cerebral cortex but also in deeper brain regions following systemic administration. Since TfR-based carriers have already been validated to a significant extent in the siRNA field, we believe we can build out our platform with relatively limited trial and error. On this basis, we have recently entered into partnerships with two companies that possess VHH-ACE technologies of this kind. First, Vectorus in France has an established track record, including a platform deal with Ionis, which supports the robustness of its technology, and it maintains a broad VHH library that we expect will enable us to identify an optimized shuttle. Key2 Brain in Sweden also has a TfR-based BBB shuttle platform and recently entered into a licensing agreement with the mid-sized pharmaceutical company Chiesi. In addition, Key2 Brain has technology to extend pharmacokinetic profiles, which gives us further expectations in terms of enhancing duration of effect. While we cannot disclose the specific targets under the terms of our agreements, we can say that they are key targets in major neurological diseases. At this stage, we are in the process of selecting optimal constructs from the VHH libraries, and we expect to obtain in vivo proof of concept data in animal models in the second half of this year.
Next, Kelly Kim from BD will discuss the key drivers and opportunities in the OliX 2.0 pipeline from a business development perspective.
As you have seen, our research team is actively advancing programs across a range of tissue types. Overall, siRNA offers a good balance between development risk and efficiency, and the modality has already been well validated through multiple commercialized products. And it is true that many other companies are actively advancing programs in this space. To further solidify our position as a leader in the field, we've put together the OliX 2.0 roadmap and are expanding our focus from liver to beyond the liver. This is closely in line with the strategic direction pursued by leading global pharmaceutical companies. The areas we are focusing on, obesity, CNS, and ocular diseases, are also the key investment priorities for Big Pharma, and we are seeing strong interest in these fields in our partner discussions. With that in mind, I would now like to turn to how we're translating these R&D achievements into real business development opportunities. Next up is our Adipose targeting program for obesity. As mentioned before, ALK7 is considered a highly promising target, with genetic evidence showing that its inhibition can reduce body fat and improve insulin sensitivity. In the obesity market, following GLP-1-based therapies from Eli Lilly and Novo Nordisk, competition to develop next-generation treatments is ramping up quickly. Major pharma companies like Roche, Pfizer, Amgen, and MSD have all announced plans to expand their presence in this space, and we too are focused on advancing our obesity drug programs. In particular, Roche's recent major deal with siRNA company Sanofi Bio has once again highlighted Big Pharma's strong interest in siRNA for obesity. While the specific target has not been disclosed, Roche has clearly stated its intention to strengthen its obesity portfolio, and given that ALK7 is included in Sanofi Bio's pipeline, many in the industry speculate that this deal may involve ALK7. Targeting ALK7 is relatively challenging with traditional modalities. For instance, small molecules need to bind to an internal pocket on the ALK7 protein, but this pocket is structurally very similar to those on ALK4 and ALK5. That makes it easy for the molecule to bind unintentionally to these other proteins, creating a high risk of off-target effects. Peptides carry the risk of hitting other pathways and often suffer from short half-lives. While antibodies might look suitable structurally, they generally can't match siRNA when it comes to target selectivity and durability, especially for tissue-specific indications like obesity and metabolic diseases. In this sense, ALK7 is not only an attractive target biologically, but also well-suited to the siRNA modality. That's one of the reasons Big Pharma sees strong appeal in our approach. At this stage, there are only a few publicly disclosed RNAi programs targeting ALK7. Arrowhead has one, but they appear to be focusing on in-house development. Companies like Alnylam and Sanogen Bio are mentioned as potential peers, though few programs have been confirmed publicly. Given Arrowhead's internal focus, there aren't many partnerable assets in the ALK7 RNAi space. For these reasons, our ALK7 program is drawing a lot of interest from multiple companies. We're receiving requests from Global Pharma for data updates and BD discussions, and we see the program as a strategic option for potential partners. We'll continue having meaningful discussions with the right partners. Let me move on to CNS, the sector really heating up in bio right now. To keep up with this trend, we are going after two-pronged development strategy. The first approach targets rapidly progressive and life-threatening diseases using intrathecal administration, while the second focuses on more chronic, slowly progressive conditions by combining siRNA with BBB, blood-brain barrier shuttle technologies. Competition for BBB shuttle technologies has ramped up in the CNS market. Major global companies like Lilly, Roche, AbbVie, and Takeda are adopting a variety of approaches, including antibody-based, TfR targeting, and lipid-based platforms to strengthen and reshape their CNS portfolios. Although recent advances have signaled that CNS therapeutics are becoming more feasible, the limited number of treatment options and validated targets make CNS an area where early-stage collaboration is possible. Unlike antibody-based therapies, siRNAs can regulate gene expression at the mRNA level and can be designed for long-lasting knockdown. In CNS today, there are a few approved Alzheimer's drugs targeting amyloid, but they usually only slow cognitive decline by around 30% and do not fundamentally stop disease progression. In many other CNS indications, treatment options remain very limited or nearly non-existent, so the unmet need is huge. Many CNS targets aren't enzymes, aren't on the cell surface, and don't have obvious binding pockets, which makes them tough to tackle with small molecules or antibodies. In this context, a long-acting approach that can suppress gene expression for weeks or months at a time, rather than relying on daily oral dosing, offers a huge advantage for patient convenience, especially in conditions like dementia. This idea that the long duration and high target selectivity of siRNA fits CNS targets really well has resonated with pharma companies, and as a result, our CNS programs are getting a lot of attention. In fact, many big pharmas already have their own BBB shuttle technologies, which gives plenty of room for partnerships in this space. We plan to continue research collaborations with local and global partners, validating a range of technologies and identifying best combinations and constructs for our platform. Through this approach, we aim to rapidly build a robust CNS pipeline around our platform, rather than focusing on individual programs, and to position ourselves as a first mover in the space. Lastly, I would like to touch on our efforts in dermatology. Although we have not spoken much about it to date, as it is one of our key growth engines, I wanted to take this opportunity to highlight it. As many of you know, we are currently running a clinical trial in androgenetic alopecia, and in connection with this, we developed a serum-type formulation, which some of you might already be familiar with. To keep it simple, we ran a pilot human study last year in Korea, and the results were positive, with encouraging responses from participants. Through this study, we saw that our siRNA technology can extend beyond therapeutics into the beauty market, and we are now engaged in active discussions with a range of companies in the cosmetics and beauty sectors. Globally, there aren't many siRNA companies that are developing and commercializing both pharmaceuticals and cosmetics in parallel. We believe our platform gives us a unique position to cover both areas, allowing us to target a much larger market in terms of technology expansion and partnership opportunities. As a new growth driver in the skin and hair area, we have also launched a program that I am sharing here for the first time. We are currently developing a candidate for alopecia areata, which is mechanistically distinct from androgenetic alopecia. This asset is designed to modulate autoimmune responses, reduce inflammation in patients, and promote hair regrowth. We are advancing stepwise through the preclinical stage and look forward to sharing more details at a later time. From a BD standpoint, autoimmune diseases have once again emerged as a major area of focus for both big pharma and derm specialty pharma, and we are already engaged in multiple discussions around this program. Yes, we're working on multiple fronts, and to me, that's exactly what makes being a platform company so powerful. That's the beauty of it. As we've always done, we're moving forward together to keep strengthening our impact in siRNA drug development and our role as an innovation leader. Like mentioned today, we're having ongoing conversations with multiple global partners, and we're focused on turning these discussions into meaningful collaborations as quickly as possible. Thank you for your continued support, and we look forward to bringing you even more exciting updates in the near future.
This is Youngjin Kim, CFO at OliX. I will walk you through OliX's financial performance for 2025 and our outlook for 2026 based on financial statements. As of year-end 2025, OliX's total assets were 195.4 billion won, up 174% year-on-year, while total liabilities were 45 billion won, down 14% year-on-year. A more detailed breakdown of assets and liabilities will be provided on the following slide. Turning to our 2025 income statement, revenue increased by 158% year-on-year to 14.7 billion won, driven by new licensing and co-development agreements, while operating expenses, mainly R&D investment, rose by approximately 23% to 44.6 billion won. As a result, operating loss was 30.0 billion won, broadly in line with 2024, and net loss, after reflecting non-operating items, narrowed to 15.7 billion won, down 61% year-on-year. Let me also briefly cover changes in equity in 2025. Starting from equity of 18.5 billion won at the beginning of the year, the issuance of 120.5 billion won in convertible preferred shares and other movements resulted in year-end equity of 150.1 billion won, representing an increase of roughly 700% and marking the highest equity level since the company's founding. Next, let me review the trends in our key profit and loss indicators over the past 7 years. As shown by the red line, OliX's revenue has shown a sustained upward trend, driven by an increase in licensing and co-development agreements. Our revenue is entirely derived from such licensing and collaboration contracts, and in 2025, we recognized portions of the upfront payments from Eli Lilly and L'Oréal, as well as milestones from Hansoh Pharma, achieving the second highest revenue level since 2023. We view 2025 as an important turning point at which meaningful revenue generation has begun in earnest, and our growth story is starting to translate into tangible sales. The green line shows operating expenses, which have been increasing in line with the expansion of our clinical pipeline, advancement of clinical stages, and development of new platforms. In 2025, clinical expenses related to OliX 104C, OliX 301A, and OliX 702A accounted for the majority of these expenses. Finally, as indicated by the gray line, net loss was the lowest since 2019. This improvement was driven by share price appreciation, which increased the valuation of put options on our shares and led to a sizable gain on derivative valuation. Next, let me explain why biotech companies like OliX, which rely on technology out-licensing as their core business model, exhibit a different earnings profile compared to companies in traditional industries. In manufacturing, for example, Direct and indirect production costs are capitalized as inventory and recognized as cost of goods sold simultaneously with revenue when products are sold, ensuring expenses and revenues are matched in the same period. Similarly, in shipbuilding and construction, for long-term custom projects, companies can recognize revenue using the percentage of completion method, provided certain criteria are met, recognizing costs as incurred while booking revenue proportional to project progress, allowing profits to be recognized progressively as the contract margin is realized. By contrast, in biotech, substantial capital is typically invested upfront in platform development, preclinical studies, and early clinical trials, with licensing deals generally occurring only after platforms are validated or meaningful clinical data are generated. Because criteria for capitalizing early-stage R&D costs are very strict, most biotech companies expense these costs immediately as incurred, then recognize licensing revenue either upfront or over time once deals are signed. This structural mismatch, where expenses precede revenues by years, makes it difficult to align costs and revenues in the same periods as in other industries, which can make financial metrics appear relatively unfavorable during the investment phase. OliX is no exception. Expenses are recognized ahead of revenue, resulting in losses through 2025. However, we view our current R&D spending as an investment that will drive future revenue generation. As shown on the previous slides, the steady increase in licensing and co-development deals and associated revenue demonstrates that our past R&D investments are successfully translating into concrete returns. Let me now review the key components of our statement of financial position. As of year-end 2025, approximately 58% of OliX assets, or 113 billion won, consisted of cash-related assets, providing solid liquidity with no concerns regarding cash burn. Turning to liabilities, 49% of our total liabilities, or 22.1 billion won, are short and long-term borrowings that are scheduled to be repaid in installments over time, and therefore have minimal impact on our liquidity position. The next largest component is contract liabilities, which represent payments received from partners that have not yet met the criteria for revenue recognition. This includes upfront payments and other consideration received under our licensing and co-development agreements, which are recognized as revenue on a deferred basis over a specified performance period. Accordingly, the 14.8 billion won balance of contract liabilities at year-end 2025 will be amortized and recognized as revenue over 2026 and subsequent periods. Lastly, let me outline OliX's key financial outlook for 2026. First is growth in operating revenue. As noted earlier, upfront payments and milestones from existing contracts are recognized over time, so a portion of the current contract liability balance will be recognized as revenue in 2026. In addition, as disclosed in the late 2025 co-development milestone announcements, research milestones may occur repeatedly over a short period, and we also anticipate sizable milestones from our licensing agreements, leading us to expect that operating revenue in 2026 will reach a record high. Turning to operating expenses, including R&D, we expect clinical costs to increase in line with the advancement of our core pipeline programs, and we also anticipate R&D spending related to platform development and preclinical activities in adipose tissue, CNS, and dual-target platforms. In 2026, we plan to execute operating expenses, including R&D, based on a disciplined selection and concentration strategy, taking into full account our expected near-term cash inflows and overall financial position. As mentioned earlier, we view our proactive R&D investments into ongoing clinical trials and platform enhancement as drivers not only of future revenue growth, but also of broader diversification in our revenue base through a larger number of licensing pipelines and platforms and an expanded partner network. We expect to maintain a very solid financial position in 2026, similar to 2025, with cash-related assets projected to increase year on year, a debt ratio that remains low compared with sector peers, and no material risk of breaching Korea securities dealers' automated quotations, monitoring criteria related to pre-tax loss relative to equity under the typical biotech capital structure. This concludes my remarks on OliX's 2025 financial performance and our outlook for 2026.
This concludes all of the presentations we have prepared for today. I would now like to ask all of our presenters to join me in thanking our audience. Thank you very much. OliX will continue to strengthen its R&D capabilities and do its utmost to deliver meaningful results in the global market. We will also further enhance our communication with shareholders and transparently shares key developments and strategies. We kindly ask for your continued interest and support for OliX. Once again, we would like to express our sincere appreciation to everyone who joined us today, and with that, we will conclude the 2026 OliX Pharmaceuticals Online IR. Thank you.